My name is Joaquin Espinosa. I work for people with Down syndrome, and I will do a few introductory remarks here. Haley, can you see my screen?
Yep.
How's the audio?
Good.
The volume is good. There's nothing I can do about the Spanglish, but if the audio is low, you let me know. Very good. Welcome to the spring of 2025 Include DCC webinar. Here's the agenda for today. I'll do a brief set of welcome remarks, and then we'll be hearing during the first hour from my dear colleagues in the Include Data Coordinating Center. Pierre Law will be talking about the Include Data Contributor Guide. Dr. Melissa Hendel will be describing strategies for management of clinical data within the Include Data Coordinating Center. And then my partner in crime, Dr. Adam Resnick, will talk about new features in the Data Hub. We'll do so within 50 minutes or so. Leave time for questions. And then at the top of the hour, we're very excited to host today our colleagues at the so-called Down Syndrome Clinical Cohort Coordinating Center, DS4C, who will be introducing the Include Collaboration for Down Syndrome Progress. After the presentation, I'll be presenting, I will wrapping up the day, with a presentation on another new initiative by the Include Project known as the Include Down Syndrome Biorepository. So we don't have today our dear project scientist, Dr. Huichin Lee, but she told me to send his greetings and to share with you some important metrics about the resource, the Include DCC website and Data Hub. If you haven't checked, if you're new to the community and you haven't checked the Include DCC website, please do. includedcc.org, loaded with many resources. And today, I have picked on Philip to demonstrate our investigator registry. This is something that we built for you, where you can learn about all the Include funding investigators, their grants, their publications. Please do check, let us know if we need to include new grants, new publications. Tons of traffic, more than 19,000 all-time visitors, more than 80,000 all-time website reviews. This is a global community of Down syndrome researchers. If you want to know more about this community, within the Hub, we have a community tab, where again, I picked on Philip today. You can find all the registered users. If you have an account there, please put a picture. Please fill out more information in your profile so people know about you and they may reach out for collaboration. We also have a forum within the website where you can see like-minded folks asking questions, trying to secure help with the use of these resources, which leads me then to the Help Center. If you need help, you can go to About, and then second from the top is our Help Center, loaded with resources that explain how to use the Data Hub, how to search data, analyze data, sorry about that, FAQs, and a large resource library. Let's talk about the Data Hub a little bit. Now, for the Data Hub, of course, you need to navigate away from the website, going to the landing page, and then sign in. We have, last time I checked, more than 1,000 registered accounts. And of course, the Data Hub, as you will hear today from Adam, is loaded with cool features for data analysis, search of data, biospecimens, and many various entry points to the large multidimensional data set of the Include Data Coordinating Center. Something that we're very proud of, that for the control access data sets, like genome sequences, for example, that require a dbGaP request, we have 36 approved requests. This is the timeline of approval since we launched the Data Hub back in early 2022. If you want to know how to apply, this needs to be done at the individual level. This cannot be done at the group level. Go again to the Help Center, look for applying for access, and there is a clear tutorial on how to request dbGaP access for a particular data set. Now, we're also very excited about the use of our cloud platform, Cavatica. You may have heard us talk about this before. You do not need to buy a supercomputer or set up your own cloud environment. You can use the cloud environment that we provide for you through the Cavatica platform. And you can request cloud credits that NIH will review for you to use the cloud. And take a moment to scan this QR code that will tell you more about it. And there are 3 tiers of cloud credits. Small amount to get introduced to the platform. It's per public projects. About $1,000. That's the next step to complete small analysis, carry out some experiments. You can do quite a bit with $1,000. And this is very easy to get, this $1,000. And then if you need more larger scale analysis, you can ask NIH for more cloud credits. I don't think they are as very difficult to get as well, but there is a review process required. And then to wrap up here, let me remind you that we host monthly office hours, the second Wednesday of every month at 12 p.m. Eastern time zone of the USA. Next one coming up on Wednesday, May 14th, 2025. So with that, I'll stop sharing and I will introduce Pierrette Law, who is a key member in the Include Data Coordinating Center, part of the data intake team, who will tell us a little bit about the Include Data Contributor Guide. Pierrette, all yours.
Okay, thank you. Can you all see the slide, or are you looking at my presenter view?
Nope, you're good.
Okay, and then you can see the hub here?
Yep.
Yes.
All right, fantastic. Sorry, move some of this stuff out of the way. So I'm Pierrette. I'm a researcher at the Linda Cernick Institute for Down Syndrome at the University of Colorado Anschutz Medical Campus. And I'm a data manager with the Include Data Coordinating Center, or DCC. A big part of my role is helping investigators, like hopefully some of you, share their data with us. So today I'll be talking about what data looks like in the Include Data Hub, how to format and submit your data, and then what we at the DCC do with it to get it ready for sharing through the portal. I'll be focusing on what we call the clinical data, which is demographic and health information about the participants in your study. So I'll start with a quick tour of some of the areas of the portal where you can see your data in action. So this is the overview of all of the studies that we have in the Data Hub and what types of data are available for them. And you can see along this side the search facets that you can use to search across the studies for various characteristics like research domain, study population, study design, etc. So this is all information or metadata that we collect from you in a structured and standardized way. So if you click on any of these links, such as this one for the Human Trisome Project, it brings up the individual study page for each of these studies. So this summary section at the top recapitulates the information that you saw on the previous study's overview. And this section can go up on the Data Hub even while you're still working on data collection. So once you have data in the Data Hub, we can show some summary statistics for you. And then down here at the bottom, we have what we call data sets, which are groupings of files. So shown here, the files are grouped by data type. And this is so we can provide you with information like data type or experimental platform, etc., that applies to all of the files in each group. And eventually, we will be providing digital object identifiers, or DOIs, for these data sets and your study so that you can link to them in your papers. So the data exploration tab also allows you to search across all the studies, but this time you're looking in the actual data. So you can search through participants for things like sex or race or co-occurring conditions. You can look through biospecimens for things like sample type or age at specimen collection. And you can look through data files for things like data type, experimental strategy, and file format. So the data that we show here in the data exploration page is what we call harmonized data that we've transformed to match the Include data model so that we can aggregate data from all of the studies. And then finally, if we go back over to the study page, this time for DS Connect, I wanted to point out an example of what we call unharmonized data. So for demographics, health history, and other tabular type data, we also share what is essentially the raw data files that contributors have submitted. And you can access these files by clicking the Analyze in Cavatica button. So portal users can download these files so that they can see what the data look like in its original form, as well as being able to use any of the data that we did not harmonize. So now I'll take a step back and talk about how we get from your data submission to what was displayed in the portal. And shown here is a summary of our data intake and processing workflow. So we start at the intake phase by working with you to collect general information about your study. And then you format and submit your data to our submission platform. We review your data and get the unharmonized files ready to be shared. And then we harmonize your data. After that, it undergoes a few more steps that I won't cover today, but then it is finally made available in the Data Hub. So now I'll go into a bit more detail about each of these steps, and I'll be referring to our Include Contributor Guide, where most of this reference information is documented. And I think Haley is going to put the link in the chat for you. So we start with the intake phase for new studies. If your study was recently funded by Include, we'll be reaching out to you. Otherwise, if you have an older Include grant or you're not Include funded, but you would like to share data with us, please email us at info@includedcc.org. So you'll start by scheduling an intro call with the DCC, and you'll fill out the official NIH project intake sheet, which is this Word document. During the intro call, we'll meet your team, get to know a bit about your research, and collect additional information about your study and data sets using another form. So the metadata model for the studies and data sets is described in the contributor guide in this tab. So this table is basically a dictionary for our data model. And we've shown here the domain, which is study or data set, the attribute, which is the data elements that we're requesting, the description, whether it's required or optional, whether there are any restrictions on the values that you can have for that attribute, and then an example. So you can see that these attributes here are the same data elements that were shown in the study pages on the portal. And this table is really just for reference because we'll be working with you pretty closely on this part of the process. So again, I'll be focusing on the demographic and clinical data today, but at this stage, we'll also find out if you have any sequencing or imaging or other data that might require access controls, and we'll guide you through a separate process for that data. And also, as I mentioned previously, this intake step can occur before you're ready to submit data, but we can still use this information to set up your study page in the Data Hub. So when you're ready to submit data to the Include DCC, please reach out to us again so we can help you with these next steps. You'll start by assembling your demographic and clinical data, which is the basic data that we need to enter your study's participants into the Data Hub. So if you're collecting data on co-occurring conditions or health history, please check the Common Data Elements tab in the Contributor Guide for this list of core conditions that were designated by the Include CDE working group. And this list is just a guideline. You're not required to submit data on all of these conditions, and we're certainly interested if you have data on other co-occurring conditions that are not listed here. So next we'll go over to this tab of the Contributor Guide that shows our data model for what we call participant and condition data. So some of the participant fields, as you saw in the portal, include sex, race, age, Down syndrome status, etc., plus a number of other fields. And then these condition fields, these are where you would list the co-occurring conditions for each participant and some other metadata like the age at which the condition was observed, or whether the condition was obtained from medical records or self-reporting. So again, these attributes are the list of elements that we need or want from your data, but we are certainly open to more data if you have it. Our data model shown here currently focuses on those core elements that we harmonize to display in the portal, but it can evolve according to the needs of our data contributors and portal users. So you can either use this table as a template to format your data, but it's okay if your format is slightly different or you have more variables that aren't described here. So in that case, you can also download a copy of this table and then use this notes column over here. Sorry, the notes column to let us know for each of these attributes whether and how your data differs from our model. So the next step is to check your data thoroughly and remove any protected health information or potentially identifiable information. So there is another tab in the contributor guide that lists the identifiers like names or dates that need to be removed from your data before you submit it. Ideally, you would only submit structured data, but if you do have free text data that is essential, please check that carefully as well. And remember that your raw data will be shared with portal users, so also be mindful of any data that might be sensitive, like interview transcripts, even if they're not technically PHI. So the next important thing is to generate a dictionary for your data. Every field in your data must be described in the dictionary in order for us to accept your submission. You can either use REDCap or some other database software to generate the dictionary for you, or there's a template in the contributor guide that can help you create one. So for every variable in your data, you would list the domain, which is usually a form or a table, the full name of the variable, which could be like a question in a survey, the corresponding column header in your data, which hopefully does not contain any spaces or punctuation, a description, whether it's required or optional, whether there is a list of permitted values for each variable, and importantly, also how you indicated missing values. So this dictionary is really important for us to use in the harmonization step, and it will be shared with portal users alongside your unharmonized data. And finally, you'll upload the data to our submission platform called Synapse. There are instructions in the contributor guide, and we'll assist you with setting up an account. So I won't go into the details of that today. You'll also go through a similar process to assemble the information about your biospecimens and data files if you have any. So the model for those components is also in the contributor guide. And so as you saw in the portal, some of the attributes that we request for biospecimens include sample type and age at collection. And then for data files, things like data type, platform, format, etc. And again, there's this notes column here where you can describe any differences between what we're requesting and your actual data. And of course, if you have any questions about any part of this process, we're happy to work with you to figure it out. So after your data has been submitted, we'll do some quality checks, including checking for ID issues like duplicates or inconsistencies. We'll make sure every field in your data has a dictionary entry, and we'll make sure that all of your biospecimens also have participant data. And at this point, we'll be ready to make your unharmonized data files and dictionaries available through the Data Hub. The last step I'll discuss today is harmonization. And this is where we'll transform those core variables of your data to match our data model so they can be aggregated with other studies and displayed in that data exploration tab of the portal. So the closer your variables in your data match our model, as we showed in the contributor guide, and the better your dictionaries and the notes that you've shared with us, the easier and quicker it will be for us to harmonize your data and release it on the portal. Also, as part of this process, we annotate the co-occurring conditions and other biomedical terms in your data, like diseases or phenotypes or medications, with terms from ontologies or standardized vocabularies. Even though that is part of our process, we're always happy to have you contribute your domain expertise to help us with this. And I won't get into all of the details of that today because Dr. Handel will be discussing harmonization and ontologies in her talk. And I also have more info about this in my webinar talk from June 2023, which is linked in the slides that we'll share after the webinar. So finally, I have this list of links and ways to provide us with feedback. So I have some links here to the Data Hub and its documentation website that Joaquin mentioned. Specifically here, I've linked to the dictionary for the fields that are displayed in the Data Exploration tab. I've also linked to the contributor guide, as well as the more technical version of the data model that we use internally. I've also listed several ways that you can interact with the DCC, either before, during, or after the data submission process. So if you're in the portal and you notice any issues, either general ones or specific to your data, you can click on this feedback tab here on the right and send us a note. And so we'll be letting you know when your data is loaded into the portal so that you can go in there and check our work. You can also email us at info@includedcc.org, or you can attend our monthly office hours. And there's a link here to download the calendar invite for that. We also conduct portal user interviews. So if you'd like to share any thoughts or requests from your experience using the portal, please email us to set up an interview. So everything I talked about today was a huge team effort, and I'd like to thank all of my colleagues at all of the institutions shown here that are part of the DCC. And this is also very much a collaboration with all of you who have either contributed data or used the Data Hub and asked us questions about our model or shared comments and feedback that have helped us improve. So thank you, and we look forward to working with you.
Thank you, Pierrette, for a very informative presentation and a great segue to the next presentation by one of the principal investigators here at the DCC, my friend, Dr. Melissa Handel. Good to see you, Melissa. The podium is yours.
It's great to be here, and I especially want to thank Pierrette for her leadership in working with the community to make your data maximally useful, and thanks so much for the great introduction. So let me see if I can get things to be shared properly here. Oh, why is it always so challenging? Slideshow. Wrong button. All right, can you see my screen?
Looking good.
Great. All right, well, it's great to be here today. I am going to spend a little bit of time kind of unpacking that last box on Pierrette's slides about how we actually harmonize the data and some of the work that we have that's ongoing so you can know what to expect in the coming months. We also welcome any contributions to these processes as well. All of the work is openly available on GitHub. And we also work with a lot of other coordinating centers that are across NIH. So we're really trying to come up with standardized processes for doing this, not only within the INCLUDE community with you all, but also across other NIH studies. So let's see if I can. So just a little bit more about that journey of a data set. Pierrette talked a lot about the things that happen on the left side of this diagram. So just to remind you, there's a lot of engagement that happens. It goes back and forth with the data submitters, but also with end users of the portal to understand what the ultimate requirements are. Very preliminary, just structuring and standardizing of the metadata as the data comes into the DCC. And then the part we're going to talk about today is really this data standardization and harmonization step. After that, the data is delivered to the portal and to researchers in a variety of different ways. I'm not going to really talk so much about that step today because we've talked about it previously, but we have a FHIR endpoint. The data is delivered to the INCLUDE Data Hub, but also harmonized data is delivered in a variety of different formats into the Cavatica workspace for researcher use. So that's what I'm going to focus on today. Oops. So in case you don't know, clinical research data turns out to be quite messy. Every individual study has its own variables, dictionaries, and data structures. And even those can change over time. And so that results in the more and more studies we have, the more and more data we have, which is wonderful, but also the larger challenge we have in standardizing information across them. Each cohort or study may have different data quality strategies. And different assumptions that are either implicit or explicit that are provided to us. There are different terminological codings or mappings, so different standardized terms that might be used or none at all. And of course, also the problem of having the same participant be participating in multiple studies, and we want to be able to, where consent allows, to be able to link those data about those individuals. And so there's really a need for a shared target schema, for harmonizing the terminologies and value sets across these different resources, for being able to link records across different studies, for sharing and standardizing and making explicit the validation and quality assurance processes, and the provenance for all of those decisions. And so this is the ultimate goal and sort of the black box of what happens in that harmonization step. I am really struggling with my new computer here. So just a little reminder, I think we've presented this slide before, but it's really important to understand some of the challenges that we're really trying to overcome in these harmonization steps. So if you look on the right, different data sources or different data contributors might have different questions in their questionnaires. For example, here where we're looking at race, one resource might have 4 values that are allowed, not reported: white, American Indian or Alaskan Native, or Black or African American, whereas another might have a larger set of terms that can be chosen. And you can see that some of those terms are the same and some of them are not. And if we want to combine these data sets, we have to know, let's say we're trying to combine these data sets to collapse them onto the top 4. We have to decide that unknown is going to map to not reported, and if so, make sure that that's really evident for the downstream users. And similarly, there might be other categories that we might want to collapse. On the other hand, we might want to go the other direction and just map those 4 onto that logger list that's on the bottom. And these decisions are made based upon maximizing research utility and the sort of likelihood of ease of standardization across many studies. So it's not to say that any one source is right or wrong, but we really just want to try to maximize the use of the data coming from the different contributors. Another problem that's a little bit more complex to describe has to do with the different ways in which we relate information. So for example, in one data source, we might not have a direct relationship between a sample metadata. So if a sample was collected on a certain day, a certain tissue, relating to a specific diagnosis. Another resource might actually have a relationship between that sample and diagnosis. And so we would need to sort of realign and reintegrate data from different parts of the data set to make sure that we can relate sample to case and diagnosis to case, and then be able to align to that sample to diagnosis relationship. And that's really important from an analytical perspective, as well as what you see in the data portal. So to do this, we use what's called the LinkML modeling language, and the data model that Perrette talked about is a LinkML model specifically designed for the INCLUDE program. And so shown over here on the left, you can see the raw cohort data that Perrette talked about, and you all have been contributing. We use the LinkML modeling language to create an INCLUDE data model. There is a specific schema representation. You can find that link at the model documentation link shown there. And in that model, we essentially create a data model with bindings to specific terminologies. And those are the terminologies that you see when you navigate the data in the portal, as well as when you are doing your analytics in Cavatica. So here's an example. So here we have some different cohorts, some different information from different cohorts A, B, and C, shown in the 3 sets of boxes. These are the raw data that we might receive from you in that intake process. And you can see that example of how race might be represented differently in yellow, You can see that in all three of these cases, we're looking at atrial septal defects, but it's being represented quite differently. In the top one, it's a true or false Boolean statement. In the second one, it's asking, of a set of congenital heart defects, which ones have been diagnosed? And that would be a checklist that could have one or zero to many answers. And then in the third one, we have some free text where it's been suggested. You can see the secundum atrial septal defect with its shortened form there. And so this is the kind of information that we have to be able to harmonize from across these different resources. And so at the bottom, you can see using our harmonica ontology annotation tool, we can actually harmonize this information to more than one race, as well as the specific conditions, the source text, and then the diagnosis and phenotypic codes that come from the Mondo diagnosis terminology and the phenotype information coming from the human phenotype ontology. And those are, in fact, the terms that you would use to navigate the data in the portal as well as in the analytical context. So this is what that looks like when you get there. Pirette also showed a little bit of this in her demo. And you can see over there, up here on the left, there's a way to study by participant identifier, but navigating by those Mondo and HPO codes down here on the left. You can also collect, check boxes for different race characteristics here. And when you do that, you'll see that same record that I just showed you. And so this is really kind of showing you how we've taken that information from the different resources, contributing those data and harmonizing them so that you can navigate to the same record here. And so this really helps us make that data maximally useful for analytical purposes as well as for cohort discovery. And so how do we actually do that? There's actually multiple steps that happen. There's the submitted data. And then, as Pirette was mentioning, we will create a derived data model from your data. And we will then check your data against your own model to make sure that there's nothing missing or something is inconsistent. Then we'll compare that against that target data model, that include data model that I mentioned earlier, and try to validate and map those data dictionaries and your data to the target model and then validate that. And then as using that harmonica tool, we will transform and annotate some of those ontology terms. There's a human in the loop in this process. And then the data would be delivered to the portal and to Cavatiga for analytics. This is not fully built. Only parts of it have been built. You've seen the parts that are functioning in the prior slides, but we're really trying to make this process more semi-automated by leveraging the data dictionaries and your data to help drive the development of the target model so we can encompass more content that can be harmonized across sources, but also to reduce the amount of human labor that's required in order to bring in more data faster, but also better. And so this is an example of, one of the ways that we're trying to approach this. And this is a little bit more experimental. And this is where we are taking essentially a set of variables from different sources. Here we're just looking at some social determinants and trying to understand which variables from which sources are the most similar, using an embedding approach to cluster so that we can say, okay, we've got 15 variables on the upper left there that relate to employment status. Those are probably good targets for harmonization because they're all very semantically related. So this can really help us prioritize and in a semi-automated fashion using LLMs, automate the harmonization. Again, there's always a human in the loop. We would never map anything without checking, but to have some of those wonderful new tools help us prioritize and make the process of the harmonization go faster. So this again is just another example of kind of how all of these pieces fit together. So over on the left, we have many different terminologies. We do that semantic encoding using that harmonica tool. We also are harmonizing phenotypic information from across different sources. And that is the sources that you then can see in the navigation in the portal. And over there on the right will help you understand how many patients do we actually have that are annotated specifically to that term. So for example, here we have abnormal heart morphology. There are 57 patients that are directly annotated to that term, but using the graph structure of the ontology, we can see that 138 patients are annotated to that term or its children. So this is really helpful for for defining your cohorts that you want to be using in your analytics, and really provides a really more sophisticated way and a more inclusive way of including additional participants in your analytics. So another experimental component we have is this idea of using a knowledge graph. And so just briefly, we can have different kinds of data coming from different places, from human data to animal models, even veterinary and other animal data that has information about genes and variants and phenotypes and diseases and treatments and environmental conditions. Using a wide variety of ontologies and data sources, we can pull these all together into a graph. And then we can bring that graph together with include data and provide that to you in the analytical environment of Cavatica. And so this is what we're working on right now. So we have over there on the left, the include data, with the kinds of components that Perrette was describing. We bring that data into an environment that creates a graph structure that allows us to align that graph structure with some of those public resources we've been collecting in the Monarch Initiative, but you could use a variety of other knowledge graphs as well. And in that way, you can see at the top, we might have the HTP study where we have the conditions labeled with Mondo and phenotypes that are related to those in the graph, that are related to the genes, that are related to the variants, all in that graph structure, and combining that HTP data with the different data that we have from the public domain in the Monarch Initiative or in any other graph that a user might want to bring, where we can bring multi-omics or pharmaceutical or clinical use cases together to be able to infer new knowledge based upon the combined data from the include cohorts together with the public knowledge that has been curated and turned into a graph. So we're really excited about some of the analytical possibilities for leveraging a knowledge graph-based approach within Cavatica. Oops. So just in a quick example, and then we'll finish up. So here's what happens when we can do that. This is the data model plus the harmonized data in a graph structure. So we happen to know that the sonic hedgehog pathway can relate to heart phenotypes and to genes, and we can infer the relationship between ciliopathies, Down syndrome, and specific genes as shown on the bottom. So for example, we might have cardiovascular phenotypes that are related to the PTEN gene, which result in facial asymmetry. But similarly, we also have ciliopathies that relate to orofaciodigital syndrome 1. And so this can kind of help us infer potential new relationships between specific genes and Down syndrome beyond just looking for the obvious suspects in terms of our trisomy genes. And so these could be many different kinds of modifier genes or protein interactions, and can really help reveal the underlying mechanisms of Down syndrome as well as biology in general. So I just wanted to thank, especially, I know we've had a small army in the DCC, but especially some of the folks shown here. Some of them come from other programs that have been really helping with this data harmonization pipeline, in particular, folks from the Biodata Catalyst team shown here. Especially want to thank Trish for the Harmonica tool and Madan for all of his knowledge graph work. And so with that, I'm not sure if we have time for questions, but I think that's my last slide.
Thank you, Melissa. We'll hear the presentation from Adam, and then hopefully before the top of the hour, we'll have a collective Q&A session. Next is Adam Resnick, contact principal investigator for the Include Data Coordinating Center, who will give us an update on new features in the hub. Adam, all yours.
Great. So thanks everybody for joining. And so today I'll review briefly, hopefully, can you guys see my screen? Okay, great.
Now we can, yeah.
Yep, great. I'll begin by going through a few slides just to highlight some of the features that we have deployed in the portal. And again, a lot of credit goes here to the St. Justine team, who's been really developing and implementing a lot of these tools and workflows within the portal. As I think you can probably tell, from both the, the presentation from Alyssa as well as Perrette and others, increasingly the complexity and combinatorial potential across studies and across data sets is continuing to increase. So what we're trying to do as part of these efforts in the portal is to increase the tools and ways that you can navigate across the data sets and both discover them and better understand how to combine them as you begin to explore those resources. Perrette highlighted this. One of the things that users can do even before logging into the portal now is to begin reviewing what data sets are available within the portal and the composition of those data sets. So one of the things we want to further empower is even before logging into the portal, beginning analyses, for anybody to begin to review those features, and I'll highlight that in a second. Increasingly, we also are expanding what is seen for each of the studies, really making the study pages that I'll show as a destination context for the team and the study, populating it with PubMed citations and other resources for each one of those studies. And then, really building on some of the really innovative components of the portal that began tackling the representation of not only participants and data files, but also biosamples and linkage to biorepositories, something that will be expanding in the coming years. Recognizing the complexity that emerges around biosamples, their management, and their representation, we added some visualization features that I'll review briefly. As users of the portal begin to explore and review components across participants, data files, and samples, these combinations of features present a challenge for many users to keep track of and to further explore and refine their cohorts. So we began adding additional visualization and interaction tools that I'll highlight briefly today. They're newly launched, so please provide feedback. We have an upset plot here that I'll highlight that allows users to begin assessing really the combinatorial representation of phenotypes across participants as you're beginning to filter them, and I'll highlight that in a second. And then two additional analytics, or the beginning of analytics tools that support comparisons and between cohorts or sets that are now stable entities within the portal. This includes something that we've further expanded on the differential gene expression context, adding a heat map representation, and then an exploration context that allows you to begin interpreting some of your safe sets of cohorts within the context of the portal. This really advances the combinatorial filter building features in the main dashboard, but by allowing you to really assess not only overlapping or intersecting cohorts, but also those that are excluded and the representation and numbers, for those combination. And I'll show that in a second. This is just an early view of that. So let me just do a couple of brief live demos around this. Hopefully they work, but just to give you a flavor for these types of workflows. So actually, let me go back and just make sure it's clear. So, even before logging on, now users can go ahead and explore the available studies and their content. And then directly from this page, you can begin your login process by clicking any one of the studies and to begin exploring that particular study or the rest of the portal features. So here I clicked on the HTTP studies. Now I am actually within the portal itself. And I can review that particular study and explore it further. And as I mentioned, we now are beginning to highlight additional publications associated with a particular study and several of the other features that Perrette already highlighted. So from the data exploration context, let's try and do a couple of explorations that begin to highlight some of the features that I demonstrated. So even before filtering to any participant or subset, this new upset plot framework begins to allow you to assess at least some of the top co-occurring frameworks. So for example, if you review any one of these, so if I'm highlighting over expressive language delay, you can see both how they occur across this distribution and then assess the combinatorics that occurs across these settings between them. So each time you highlight any one of these, it'll give you the prioritized assessment of that particular selection, like hyperthyroidism in this context, and then highlight how those occur and distribution-wise. Obviously, each one of these upset plots essentially shows the join across these contexts. So this is really a rapid way to assess within the studies or within that filtered content that you're reviewing this co-occurrence context that can then additionally drive the way you might want to query and assemble cohorts. So let's try doing that right now. So let's begin by, for example, choosing celiac disease as a context. As you notice, the minute I select that feature, everything down below changes, including all the associated graphs that were there already. But now you can also see within this celiac disease positive framework the distribution of co-occurring conditions in that context. So here's the celiac context, which all of the participants that are now selected will have, in part because of the selection feature that I've already put into the filter context, but then obviously you can see that distribution setting for co-occurrence. We can then also still do, besides that fast query, additional features, and again, subdivide this. So let's add another context based on the features we noticed. So let's look at the abnormality of the let's see, endocrine system, as a context. Oops, hold on, let me check. Do that. In part driven by some of these features that are overlapping in this context from the upset plot. Many of you may have begun doing this in the past, but may have not yet utilized the context of creating saved filters around either participants, biosamples, or data files. So let me just review this with you now so that we can show and highlight how these can then be further explored with some of the new analysis features. So here I selected a participant context, and I can create a saved participant set. So I'm going to save a new set. This will save it, this particular set as a context. Now let's create a couple other queries. So we have some additional features to explore. So here I did a comparison by assessing phenotypes. I'll quickly do, for example, let's do by data file. So I'll choose a data category. So here I'm choosing all participants that have transcriptomics. Let me go ahead and create another saved participant set for here. And at the same time, let's go ahead and do a biosample set. So in the same way that I save the participant set, I can also save all samples that have transcriptomics in this context, but I can further filter them by, for example, biosamples and their availability. So here I'll choose only samples that are available, right? So here I already combined across the availability of transcriptomics and sample availability to create another biosample set as a context. And you can obviously modify the annotation. You can obviously also intersect cohorts, as you might have seen before. And here, these are participants with an abnormality. Let's expand this a little bit more and actually make it an or instead of an and. And so here, this is the intersection of participants with these features and samples that have transcriptomic and are available. So you can already see this complexity that is emerging. And again, let's create another set of both participants and samples to review. I won't go through variants, which is another context where you can save sets. Let me do biosamples again. And recall, we demonstrated this in a previous presentation. You can also request samples by these features, but let's go ahead and just save these for now. All right. So I've now created a number of different saved sets, and these are saved and are available for you to review within the context of your dashboard. So here you can see the saved sets of both participants or biosamples. So anytime you return to this context, they're available, and you can directly explore these by clicking on them whenever you return in the future into the portal environment. But let me highlight some of the analysis features as well that are now new features, and I'll try and finalize these fairly quickly. First, we've had an expression comparator view for some time, but are now adding additional components into this context. So let me just remind you how this works. So here I'm just going to choose a gene, right? You can choose any gene and immediately assess the expression differences. You can also actually select a set here. So this is another area where you can actually select samples. So I'm going to select this sample, right? So I selected the samples that have high expression, and actually save this as additional set. Let's try that. So I'll do high. Okay. And I'll show you why that's meaningful. Besides comparing within any one gene, the additional feature we've now added is a heat map. And so I'm just going to select a set of genes, but you can have any set of genes of interest on your side. And you can just upload that set of genes. And once you upload them, you immediately see a heat map essentially displaying or ranking the expression for genes across this cohort as a context within the T21 context. So this is another new feature that just recently deployed. All right, so let's end, because I think I'm a couple minutes over time, with the set operations. So recall we had created a number of different sets for both participants and biosamples, and those sets can really represent that body of work that the user has done in crafting that combination of features in the data exploration context. But this now begins allowing you to further explore these in meaningful ways. So here I chose, let's just choose these two sets that I predefined, and you can immediately see not only the overlapping context, which is something that you can fairly easily do in the data exploration context, but now you can also see the exclusionary setting or the numbers of participants that in this context that are represented by the non-overlapping context for any cohort that you now have defined. We'll be building additional features and click out from this feature, but this is just actually the first day we have this launched for exploring essentially the overlapping context based on different features that you might have defined across these resources. So I'm going to select biosamples, and recall I just selected the high interferon receptor expressing. I can also add biospecimens that are available and the abnormality of the thyroid and compare these. And again, you can start seeing the biospecimen overlap context for these cohorts. Let me just finalize on the biospecimen view, and then I will complete. So for any view for a biospecimen context, this can be extensive and large. And historically, because these biospecimens have relationship to each other, and because you're trying to potentially compose requests, it's sometimes challenging to combine and review the context of each of those biospecimens. So now we've added additional views. So all you have to do is just click on any biosample. So here's that biosample. And you can further understand the relationship between that biosample, in this context, plasma, and any of its derivatives as a context and their availability. So here you can see this hierarchical view of the relationship and processing of these samples in ways that can then be driven for a request process moving forward. So hopefully that gives you a lens on some of these new features. And notice that there, for any of these pages, there's a little feedback button. Please utilize it as you begin exploring these new features. We know it takes time for users to get accustomed to these workflows, and we know that we are creating multiple different ways for a user to end up at a certain cohort. But in many ways, this is to empower the users to intersect and view the cohorts they're creating from a number of different perspectives as the larger cohorts begin to accumulate within the portal. So with that, I'll stop and look forward to questions and comments.
Thank you, Adam, for highlighting some of the newest and meanest features in the Data Hub. Yeah, we have about 5 minutes for questions. And there's a question in the chat on a very relevant topic that is very, very timely, I would say, for the Data Coordinating Center. So Jean Ling is asking how we call in chromosome 21 variants. And I don't know, Adam, if you want to take on that one, we can call on Matt Galbraith to address that. Who wants to take that one?
Yeah, if Matt's on, I'm happy to have him lead this.
I'm here. Yeah, it's a really good question. Currently, the variant calls for chromosome 21 are not correct. They're called in the diploid mode using the GATK pipeline. We have a couple of different options in the works to deal with those, and we intend to recall them all correctly, or at least in triploid mode with appropriate metrics as we vet things so that they can be used appropriately. But right now, you would have to call them yourself in triploid mode or some other appropriate way of arriving at the correct genotype calls.
Yeah, so to Matt's point, right now, we're calling it essentially with a standard GATK pipeline across, all chromosomes, across all cohorts, but are in the process of actually, as Matt highlighted, to applying that triplicate mode framework. So it's set for chromosome 21. We can have that specific view and lens on that framework. And to our knowledge, this will be the first time that there's any portal that's supporting that type of lens on the variant search.
The other thing that, as part of that we're likely to do is to do a joint calling across a larger fraction of the cohort, right? We have had several different tranches of whole genome sequencing go in here at the moment, and there isn't joint calling across all of those yet. So that may be of advantage to a number of people as well.
Ginley, you're asking about a timeline. This has been identified last week as a top priority for our team. So I don't want to commit to a date, but it's been floated up to the top. Any other questions for any of the presenters of the first hour? Monitoring the chat here is quiet. Please feel free to add your questions as the presentations are going, and we can address this.
Well, let me just reiterate also that, again, as you begin engaging the portal, make sure to use that feedback button. You can ask questions, clarify, identify issues, and we'll communicate with you on those.
Very good. And I see either Kathleen or David would like to have a question here. And yeah, feel free to unmute yourself. Kathleen and David Egan joining us.
Okay. So I noticed that you used the term participant for, I imagine that's the person with Down syndrome. That's participant. That's right?
Correct. We do have some participants without Down syndrome as well.
Ah, okay. So do you identify them as such or not?
So you have a way of filtering. We're using the acronyms D21 and T21 for without and with Down syndrome. D21 as in 2 copies of chromosome 21, and T21.
Okay.
That's the how we can filter for those with versus without Down syndrome.
In one of the presentation, there was a slide that had patient in it. So I
It
was
is.
wondering
That may
if,
be, thank you, Kathleen. That we don't use the word patient for people with Down syndrome, but it's used more generically when we are talking about biomedical research more broadly, right?
okay,
So you
because
may
I
have
remember David had mentioned at one point at the NIH Advisory Council that don't call us patients, we are participants in research. But anyway, the other question that I have is more, so when the researchers send their data, like I saw categories like intellectual delay, expressive delay, intellectual disabilities. Those are different measures than the medical measure that says the person has a VSD or the person has celiac. That's kind of an objective data point. But when you say expressive delay, maybe different researchers measure the delay with different tools. And intellectual disabilities, the measures of intellectual disabilities are compared to the normal quotient of people, you know, like the IQ test. And so people who with Down syndrome sometimes appear as 4 years old or 8 years old. So I was wondering if there is a certain harmonization or validation of what tools are used to determine if that person has this delay or what intellectual capacity they have. I'm worried about the medical tools, I imagine, are more harmonized and validated, but the psychological ones are not necessarily.
very good.
So it's
So,
just a question. But anyway, I want to say wonderful, congratulations. You've done a great
Kathleen.
job.
You're always,
Thank you so
always
much.
very supportive of our work. Let me very briefly before we run out of time, we need to get to the presentation. I want to address the comment about how do we arrive to a common term for, say, intellectual disability or delay. As you just well explained, different studies may be doing different measurements with different instruments. Then that gets mapped to a common term in either the ontology vocabulary, the Mondo vocabulary, or the HPO vocabulary. Now, in the study pages, if you want to get deeper to find out how was that delayed, as you can find the data dictionary and see if they use this test or that test, what cutoff, right? So there's a harmonized representation of primary data. If you want to see how the primary data was collected, that can be found in the study pages and the data dictionaries.
And then all of the data comes from researchers, not from the DS Connect, where the parents say something.
There is a mix of sources. There is the S-Connect data, there is data from clinics, through ICD code. It's a multitude of data streams.
Okay. Thank you so much.
Yeah, good to see you and David, and thank you again for
Thank you.
participating. Very good. We'll transition now to the second half of the webinar. And as I was saying earlier during the introduction, we had the pleasure today to welcome and host our colleagues from the newly minted Down Syndrome Clinical Cohort Coordinating Center, also known as DS4C, and that'll be Jessica and Tracy here. Jessica, the podium is yours.
Hey, thanks so much, Joaquin. I am assuming you see the right version of my slides, and I'll keep going unless someone tells me different. But thank you so much. We're really excited to talk about the INCLUDE collaboration for Down syndrome progress today. Many of you are very familiar with INCLUDE, and I'm not going to spend much time explaining INCLUDE to you, other than to note something that's been a really big part of INCLUDE's plans from the very beginning is this component 2, to build a large cohort of individuals with Down syndrome for comprehensive analysis and biomarker evaluation. And that's exactly what the collaboration for Down syndrome progress has just been funded to do. So I'm going to give you a quick overview of the CDP. We do have some acronyms, so I'm going to do my best to explain what all the components are. But to give a broad view about what the CDP aims to do is we're going to address component 2. We're going to develop a cohort of individuals with Down syndrome from across the lifespan, from birth to adulthood, collect deep phenotypic data and link that data to biospecimens to enable very detailed deep phenotypic longitudinal data analyses to help stimulate research in the co-occurring conditions in Down syndrome, to help improve our understanding of the natural history, to help better understand the frequency and variability of co-occurring conditions, to understand the heterogeneity that we see in Down syndrome, to understand the impact of different factors, environmental and molecular factors on outcomes in Down syndrome, and to help understand the implications of therapeutics across the lifespan. What we're going to be doing as part of the CDP is we're going to facilitate standardized data collection. So what we've been doing is developing what we're calling a common protocol that different sites that are funded across the country and in Latin America are going to be implementing this common protocol to facilitate standardized and comprehensive deep phenotyping and biospecimen collection. We're also, as part of this common protocol, not only aiming to get longitudinal data, but also aiming to collect a bunch of different types of data: demographic, clinical, lab, biospecimen, and imaging data. We also plan and have been implementing strategies to make sure we have a strong engagement with the community. We plan to do significant outreach, recruitment, and ongoing engagement with the community advisory boards. We are aiming for inclusive recruitment and enrollment options, plan to do focus groups and other community partnership activities. And overall, we just plan to enable transformative research in Down syndrome. And all the data generated by the CDP will be deposited into the INCLUDE DCC and available to the research community via the Data Hub. So the CDP is one addition to the existing INCLUDE ecosystem, which many of you are familiar with. So we have DS Connect, the registry, as you're very familiar, the DCC and Data Hub, the ABCDS for the Alzheimer's Biomarker Consortium, and then the CDP is just the latest component of this INCLUDE ecosystem. How we've organized the CDP is we have the Down Syndrome Clinical Cohort Coordinating Center, which we're calling the 4C. That's what Tracy and I lead. The 4C is essentially aimed with facilitating collaboration and communication across the CDP. We'll be organizing the data collection and ingestion and deposition into the DCC for analysis through the Data Hub. We'll be centralizing the outreach and engagement efforts across the CDP. And our biggest activity so far has been facilitating the development of the common protocol. And to do this, we've really engaged some of the other components of the CDP, including the newly funded DS Biorepository, where all the biospecimens are going to be collected and processed. We've also have a community advisory board that we've engaged to get feedback on the study design, from recruitment to data collection. And we have 5 research groups, clinical research sites, or CRS, that have been funded to help with recruitment and that data collection. And I'll say more about these in a bit. These are the names of the studies, and the description of the names of the studies are here. But essentially, there are 5 research teams that are spread across the United States and Latin America that have their own research aims, but are also committed to implementing the common protocol for this standardized data collection. And so we've all been working as a cohesive and exciting collective unit to help develop this common protocol. And again, all that data will be submitted to the DCC for availability to the Data Hub and stimulate research and the larger research community. Here's who we are at the 4C. Tracy and I are leading up this effort. We're providing content experts to help inform the development of the common protocol. We have a CAB, which I'll talk about in a bit. Within the 4C, we have 4 cores, and we're split across RTI, Research Triangle Institute in North Carolina, and Emory University in Atlanta, Georgia. Meg Crawford and I lead the administrative core to help facilitate all the administrative and logistical aspects of the CDP. Also at RTI, we have the data management core, including Kate Burdikin and Jenny Amon at RTI. Emory hosts the cohort core. Tracy's been leading the Herculean effort to help develop the common protocol. So she's leading the cohort core, and then Emily Allen, also at Emory, is leading up our outreach core. To come back to those 5 research teams that have been funded to help with the data collection for the CDP, here's a general overview of the research study names, who's leading those, what the general goals of the research study are. The biggest takeaway is that the funded studies represent a broad range of recruitment ages, again, from birth to into adulthood. And collectively, we're planning on recruiting over 3,000 participants into the CDP. Here's a map of all the existing CRS recruitment sites. So they're quite distributed across the United States. And very excitedly, we have pretty broad recruitment across Latin America to the HTP LAN study. So to get into the common protocol that we're developing, I wanted to say a bit about the process and kind of how we're going about developing the common protocol. We've developed 6 working groups that are each assigned a different domain of the common protocol. This helps us to streamline the expertise, to focus on specific aspects of the common protocol. So we really want to leverage existing technology to promote data capture through the remote digital health. So we have a remote digital health work group. We have a work group that's focused on collection of biospecimen and generation of omics. We have our demographic, socioeconomic status, social determinants of health and environment working group. We have the assessment data working group, which has been tasked with the very big task of identifying the neurocognitive and behavioral and other assessments to administer through the CDP. We have a medical data and electronic health record working group, and then the consent and assent working group. And each of the 6 working groups, we have 2 co-chairs. We've tried to really facilitate collaboration across the CDP by having strong representation across both the 4C as well as the 5 funded clinical research sites, the biorepository. And within the membership of all the working groups, we have made sure that we also have representation across all the CDP components as well as the DCC. There's a little bit more description about what the working groups are collecting. So again, for the remote digital health, we're really trying to take advantage of existing technology with regards to wearables and other data collection devices that can be done remotely. The omics biospecimen working group is focused on identifying what biospecimens we can collect, as well as what omics data we can generate from those biospecimens. The demographics group, they're going to be the ones identifying the best methods for generating a GUID so that all the data can be aligned across both the CRS sites in case people participate in more than one study, as well as potentially with existing data in the DCC, but also collecting other data related to demographics and environmental exposures. Again, the assessment data working group is tasked with identifying what cognitive, behavioral, and skill set assessments we can administer with relevant assessments that can be given across the age range. Medical data in EHR working group is focused on collecting data, the clinical and medical data, both from the EHR and through a clinical visit. And then the consent and assent working group is tasked with developing consent as well as participant engagement activities to facilitate consent, such as videos and storybooks. Most importantly, when we started developing the common protocol, when we got funded back in the fall, we didn't have to start from scratch. There's a lot of resources that we leveraged in informing the development of the common protocol. that we made all the work groups, provided all these resources to the work groups as a starting point. Importantly, again, all the clinical research sites had their own specific research aims. And one thing we really wanted to do is to help them reach their aims through the Common Protocol. There are some really important questions that these sites aimed to answer. And so addressing as many of their aims as we could in the Common Protocol was a priority for us. We also wanted to be aligned with other existing efforts, such as the ABCDS. So we looked at their protocol to see where we could align with their protocol to help inform those kinds of assessments across the lifespan, given their focus on adults age 25 and older. We've also been involved in prior efforts like the Trisomy 21 Research Society existing cohorts database that looks at existing cohorts of individuals with Down syndrome and see what measures have been used in the past and facilitate the use of those measures to be aligned with other existing data sets as much as possible. There have been ongoing INCLUDE workshops that INCLUDE has facilitated. Like I said, they've been planning for this initiative for a long time. And so there's been a lot of planning meetings to identify what data elements should be included in the Common Protocol. And so a lot of those decisions have been considered in developing the Common Protocol. A lot of us have been involved in other Down syndrome projects like DS360 and have contributed our experience and others' experience in those studies and what data elements are captured in those, as well as aligning as much as possible with the DCC CDEs. There's also a lot of other factors that we have needed to take into account in creating the Common Protocol. You know, it's really a great opportunity to do deep phenotyping, but it's important also to balance that with having a feasible protocol for participants to complete and taking into account the time commitment and the potential burden of doing something that's just a little too lengthy. So that's really something that we tried to do. Of course, we want to throw everything into it and capture as much data as possible, but at the same time, we want to make it approachable and feasible for participants to do. Another thing that we've had to take into account is looking at how the data collection points need to be adjusted across the lifespan. Obviously, when we're thinking about cognitive measures, we can't give the same measure to someone who's age 2 as we do to someone who's age 20. But trying to find comparable measures that we can use to track certain outcomes across the lifespan has been a really big goal in developing the Common Protocol. We've tried to leverage as much as possible available and validated measures. We've also identified important data that we would like to capture, but maybe in that data capture, it's a little too expensive or not all sites have the capability of doing that. I'll show this in an upcoming slide, but an example of that is sleep study. Sleep apnea is a really important outcome to understand in Down syndrome. And so we really wanted to include sleep studies as part of the CDP Common Protocol, but as you're aware, it's expensive. And doing that on all 3,000 participants just wasn't going to be feasible. And so trying to identify a way that we can still include that data capture as part of the CDP in a way that's affordable and approachable and feasible while still generating important data has been one of our goals. And of course, we want to make sure that we're ensuring participation that's inclusive of everyone, including a range of functional ability, age, geographic location, et cetera. So here's where we are with regards to the Common Protocol. So we have a set of data that we're planning to target in everyone that enrolls in the CDP, including assessments and questionnaires, collection of medical and other clinical data, collection of biospecimens. But then we also have this kind of what we're calling the subsample data collection of in a subset of people who are enrolled in the CDP. We plan to do sleep studies. We plan to provide wearables like actigraphs to track activity, as well as do brain imaging. But again, only in a subset. But it is included as part of the Common Protocol. In administering the Common Protocol, we actually see several different potential pathways and participation in the Common Protocol. Again, we wanted to make sure that the Common Protocol was as accessible and inclusive as possible. So what we see is engagement of a potential participant in the CDP, including initiation of consent, creating a GUID. Then we start to begin the data collection. And we want to encourage the in-person visit collection as much as possible so we can get that important assessment data and clinical data that can only be done in person. But at the same time, we understand not everyone can travel and do that. So we'll aim to collect in-person visit collection as well as some remote collection like self-report questionnaire data. But if they can't do the in-person visit, we still want to include them in the CDP. And so in that case, we will focus on administering them the remote data collection aspects of the Common Protocol. If they are enrolled at a site that's collecting what we're calling the subset data, sleep, physical activity, and brain imaging, they'll be asked if they're willing to participate in that aspect of the Common Protocol. And if they do, we will collect that data. And then for all participants, remote or what we're calling hybrid of in-person and remote data collection, we'll facilitate longitudinal follow-up to collect data points across the lifespan of the CDP. And we also aim to collect all data possible for the Common Protocol, but at the same time, we understand that there may be limitations in what data elements people can complete, such as all the cognitive assessments, for example. And so though we'll aim to anticipate barriers to data completion and identify ways to overcome those barriers, we also understand that that might not always be feasible. And so we'll collect as much data from the Common Protocol as we can, but also identify chances for recollection for any missed data elements as we follow them over time. We want to make sure that the data is collected in a standardized process. So as part of that, we're going to develop SOPs. Each site is going to be administered an electronic data collection program. We're going to facilitate centralized training of implementation of the Common Protocol, monitor the data quality over time, and set up robust communication channels to support the Common Protocol implementation. And then here's just a general overview of where we are in the CDP. So we're still in the first year. We're still developing the Common Protocol. In the middle of year one, we plan to have the Common Protocol finalized and submitted for IRB approval so that in year two, which starts this fall, we'll all be ready for data collection and recruitment. So that's kind of where we are and getting ramped up for the CDP. For data elements that we still think are important but aren't necessarily included in the Common Protocol, we'll think about how to leverage this existing infrastructure and network that we've created with the CDP to perform ancillary studies, which will be another way to perform standardized data collection and deposition in the DCC to make that data available for interesting research questions that may not be addressed in the Common Protocol. And of course, a really big part of what we want to do with the CDP is make sure we have good community outreach and engagement. And so we have at the 4C a community advisory board with different representatives and advocacy individuals. And then each of the 5 clinical research sites has their own CAB. And on this overarching 4C CAB, we have one of those representatives as part of the overarching CAB for the overall program. And our general plan for community engagement is networking in community groups, including engaging with local and national organizations. Again, we have our CAB both for the 4C as well for all of the individual research studies. We're working on getting community feedback on the Common Protocol design and implementation, including recruitment and engagement, the Common Protocol and incentives for participation, whether that be monetary or other types of resources that participants and their families would like access to. We also will have a centralized call center for family sites and health professionals to answer any questions that people may have about the CDP or the Common Protocol, and providing family-friendly resources, attending local conferences and webinars, newsletters and infographics to share any research findings, as well as doing what we're doing now of promoting Include in the CDP at the national level. And so that's all I had today. Thank you.
Very good. Thank you, Jessica. And we do have 5 minutes for questions. Monitoring the chat here. Is there anything, Tracy, that you would like to add?
No, we're just super excited. I think all of the working groups are making some great progress. So hopefully we will have everything finalized in the next few months.
Very good. Yeah. Lots of progress being made, and yeah, lots of excitement as well. Very good. I can get started with the biobank presentation, and please do enter questions either for Jessica, Tracy, or the next presentation, and hopefully we'll have time before the top of the hour to take more questions. Very good. And we'll wrap up the day today with a presentation about the Include Down Syndrome biorepository that Jessica highlighted in one of the slides there as one of the key components of the CDP. And I'll be presenting today on behalf of a much larger team. So this is one of the components of the CDP, as Jessica described. I serve as the contact principal investigator, and the other PIs are Angela Raczubinski and Mal Galbraith from the Cernic Institute, and Elizabeth Head from the University of California, Irvine. And we have many, many wonderful co-investigators and other senior key personnel highlighted here: Laura, Audrey, Brian, Erika, and many, many more. This is like all of these grants that we're talking about, the DCC, the CDP, and others. These are cooperative agreements, meaning NIH is strongly involved in all decisions and management of the activities. In our case, the biorepository is a grant from the Include Project through the National Institute on Aging, and our program officers and scientists are Lori Ryan, Nandini Arunkumar, Erika Tarver, and we also have support from the Office of the Director, Nikila, Kayla, Aisa, and others as well. So our mission is to catalyze discoveries that benefit people with Down syndrome by enabling transformative biospecimen collection, processing, distribution, and analysis. And here are some of our guiding principles: service, innovation, collaboration, and stewardship. Of course, we're here to serve first and foremost people with Down syndrome, but as a resource funded by NIH, our clientele would be biospecimen collectors, studies who are interested in collecting biospecimens as part of the Include project. So we have multiple stakeholders. Innovation, we believe that people with Down syndrome deserve the most cutting-edge, state-of-the-art biospecimen management and analysis platforms equivalent to what is being done for the study of other medical conditions. Collaboration, this is key. It would take a number of stakeholders. It will take a village to make to do this right. Not only the families, the self-advocates that provide the biospecimens, but also the various research studies, NIH, downstream requesters, users of those biospecimens. And then, of course, the stewardship. This will be precious biospecimens that people with Down syndrome will be contributing to the biorepository. We need to make sure that they are used strategically, wisely, and also stewardship of the taxpayers' dollars. This is not an inexpensive enterprise. It takes quite a bit of resources to do this right, so we have to be mindful of that as well. The goals for the biorepository really are one and the same that the goals for the other components and for the Include project overall. We're in the business of elongating the lifespan of all people with Down syndrome and improving the health of all people with Down syndrome. What do we mean by elongating the lifespan? Life expectancy in the US in the general populations is in the late 70s. for people with Down syndrome in the US and other developed countries is in the late 50s. People with Down syndrome are missing 20 years. That's the object of our study. And then, although great progress has been made in the clinical management of Down syndrome, people with Down syndrome are still affected disproportionately by a number of co-occurring conditions. So, of course, at the end of the day, it all arises from that extra copy of chromosome 21, trisomy 21, leading to those common yet highly variable traits: stunted growth, neurodevelopmental delays, early aging. Now, we know that people with Down syndrome have a different clinical risk profile. We can de-emphasize the study of some conditions that are less common in Down syndrome, like cancer, atherosclerosis, hypertension, and we must prioritize the study of co-occurring conditions that are more common in this population: respiratory illness, autoimmunity, Alzheimer's disease, various vision and hearing issues, and more. So in order to deliver on this mission, we must study the co-occurring conditions of Down syndrome. So the scientific inquiry is very clear. We know what causes Down syndrome. Down syndrome is a genetic condition. It's not fluoride in the water. It's not vaccines. It's not poor parenting. It's an extra copy of 200 genes or so encoded in this small piece of DNA. So we need to find out which of these genes are causing which features of Down syndrome. And if we could figure that out, we could perhaps come up with strategies to counteract the undesired effects of chromosome and gene triplication in people with Down syndrome. This is doable, okay? This is achievable. Let me give you a couple examples. We know now that it's the APP gene, the so-called amyloid precursor protein gene, that is driving this high risk of Alzheimer's disease at an earlier age in people with Down syndrome. APP encodes for a protein that gets cleared, giving rise to the so-called beta amyloid. When that beta amyloid accumulates in the brain, triggers a pathological cascade that leads to Alzheimer's disease. There may be other genes on chromosome 21 that may also contribute, but we know for a fact, the science is very solid, that APP carries the bulk of the phenotype. And based on this understanding, now there are many clinical trials targeting APP in people with Down syndrome, APP vaccines, monoclonal antibodies, targeting APP, also antisense oligonucleotides to lower the dose of APP. And this, of course, allows the development of biomarkers of disease progression. Let me give you another example. We know there are 4 interferon receptors on chromosome 21, the 2 type 1 receptors, one each of the type 2 and type 3 receptors. This leads to an elevated interferon response, an elevated innate immune activation, increased JAK-STAT signaling, that's the signaling cascade employed by the interferon pathway, overexpression of these immune genes, leading to a state of chronic immune activation and inflammation that contributes to clinical and developmental hallmarks of Down syndrome. In mouse models, it's very clear that triplication of this gene cluster contributes to multiple hallmarks of Down syndrome. With this understanding, now the INCLUDE project is funding multiple clinical trials to test the safety and efficacy of JAK inhibitors for different clinical endpoints, okay? So this is totally achievable. Just like we figured that APP triplication can drive Alzheimer's disease, leading to anti-APP therapies, interferon receptor triplication leads to immune hyperactivity, leading to testing of immunotherapies. Now we must find what other gene products are driving all the pathological processes so that we may illuminate other therapeutic opportunities. So how do we do that? How do we do that? Again, we agree on the notion that Down syndrome is a condition of dysregulated gene expression. And those genes, when overexpressed, will have, of course, effects on all the genes of the transcriptome, on the protein, on the metabolome, then impacting cells, tissues, and organs, leading to the presentation of Down syndrome. So we need to collect data sets that can inform about the molecular genetics of Down syndrome. Data on DNA, RNA, protein, and other molecular data sets that can tell us about gene function. So how are we going to do this? Well, biospecimens can be analyzed with a number of so-called omics technologies that allow high throughput, larger scale analysis of the effects of the trisomy on gene expression programs, the transcriptome, on chromatin architecture and organization, the epigenome, on gene function through functional genome experiments such as short hairpin or CRISPR screens, analysis of the protein, the metabolome, the immune cell repertoire, so on and so forth. Clearly, this will take multidimensional biobanks that can lead to multimodal data sets for collaborative discoveries of multidimensional impact. As it was mentioned earlier, we are following guidance from previous working groups. There was an Include Biospecimens working group that prioritized all these wonderful data sets from those that are likely of the highest value in a universe of finite resources. Analysis of the genome, analysis of the transcriptome, the plasma proteome, and if resources and effort allows, other omics data sets, the epigenome or a microbiome, generation of iPSCs. We'll talk more about these, immune maps, autoantibodies, and other wonderful data sets. Now, with that being said, we need to acknowledge the challenges so that we can come out with solutions to overcome these challenges. One is that trisomy 21 affects all major organ systems. Unlike some colleagues across the hall that may be studying a condition that affects only one organ system, we don't have that good fortune. You know, we need to study the impacts of trisomy 21 across all organ systems. Trisomy 21 causes different effects at different life stages. It's a chronic condition from the moment of conception all the way to old age. And also we need to acknowledge that Down syndrome actually is a bit of a bucket word. Down syndrome can present in many different ways, highly heterogeneous presentation. So how could the biospecimens then help us address these challenges? Again, let me unpack this a little bit. The chromosome does something different at every organ system, you know, affects the CNS, leading to neurodevelopmental, psychiatric, and neurological phenotypes. It may affect the cardiovascular system. We know, for example, it causes congenital heart defects, obstructive sleep apnea, high propensity to respiratory tract infections. It affects the immune system, leading to an autoimmune prone state. Of course, it also affects the musculoskeletal system, leading to other phenotypes. So we need to find a way to use biospecimens to fuel investigations of trisomy 21 across these organ systems. So how is this done? Well, there are biospecimen types and omics approaches that can educate us about the impact of trisomy 21 in different organ systems. For example, the plasma proteome, the plasma metabolome, they carry information on the function of virtually every organ system. Every organ has a proteomic, a metabolomic footprint in the blood, right? And this is obvious to anybody who went to the doctor and gave blood for a test and another. What are doctors doing with that blood? Well, they may be looking at markers of liver dysfunction, liver enzymes, right? They may be looking at metabolites that inform about kidney function. If you have an immunological condition, doctors may be looking at neurofilament light in the plasma that informs about levels of neurodegeneration. Let me give an example of how this works real time to make discoveries. This is a paper published a couple years ago measuring neurofilament light in 400 research participants in the Human Trisome Project. And as you can see, neurofilament light is elevated in Down syndrome, T21, at every age bracket. So people with Down syndrome have elevated biomarkers of neurodegeneration. With that being said, there's always a range, right, in Down syndrome. So to understand that viability, we did a matching analysis of the plasma proteome, measure 5,000 proteins with the SomaScan proteomics platform, and look for proteins that have positive or negative associations with neurofilament light. The most positively correlated protein starts with IGF, IGF-BP2. The most negatively correlated protein, IGF-1. This makes sense. IGF-BP2 is the repressor of IGF-1, the insulin-like growth factor. Now, IGF-1 works in collaboration with IGF-ALS and IGF-BP3. All three proteins are negatively correlated with NFL. Let me say that again. Per every unit of increase in the marker of neurodegeneration, the repressor of IGF-BP2 goes up, IGF-1 goes down. So this statement is true. Low IGF-1 signaling is the top proteomic signature associated with high levels of NFL in Down syndrome. What is going on? IGF-1 is part of the growth hormone signaling pathway, is produced by the liver downstream of growth hormone. And then it binds to surface receptors that lead to cellular proliferation and organismal growth. IGF-1 is a master regulator of human development, including brain development and neurogenesis, right? Now, all three factors are made by the liver, right? Suggesting that there may be something off with the liver in Down syndrome. These are the data points, 400 research participants in gray, levels of IGF-1 in the general population, going up in the first 2 decades of life, then dropping down. That's true for the 3 subunits of the complex. So the pituitary seems to be doing the right amount of growth hormone, but the liver seems is impaired in making the right amount of the IGF-1 complex, leading to the question of, how is the liver in Down syndrome? And also there could be a therapeutic opportunity here to use either growth hormone or IGF-1 supplementation in Down syndrome. So now it turns out you have another omics data set that you can interrogate, in this case, metabolomics. And what do you see at the top of the charts? Multiple bile acids clearly upregulated in Down syndrome. This is just one example, thyritocholic acid, highly elevated. High bile acids are a biomarker of liver stress, liver dysfunction in Down syndrome. So why are bile acids elevated in Down syndrome? Let me take a little detour here and tell you that if you collect biospecimens correctly, you can reprogram cells into induced pluripotent stem cells, which then can be differentiated into a multitude of cell types. So this allows you to study the impact of trisomy 21 in contexts that will be very hard to biopsy: bone cells, blood cells, various other organs. So Lauren Dan, Brian Yemaya, and other colleagues then took iPSCs with trisomy 21, differentiate them into hepatocytes, and sure enough, they documented multiple events indicative of liver stress or liver dysfunction. These hepatocytes make less albumin. Hypoalbuminemia is a hallmark of liver stress. These hepatocytes accumulate more fat droplets. And when they do an RNA-seq experiment, they see elevation of many enzymes, genes involved in bile acid metabolism, and sure enough, in the supernatant of the Petri dish, there is increased bile acid. So this is a clear example how biospecimens properly analyzed can lead to a sequence of experiments and discoveries. We also must acknowledge another challenge, and that is that the chromosome is doing different things at different life stages, and that manifests in the clinical presentation of Down syndrome. Congenital heart defects, of course, are issue for newborns, but then the heart seems to be doing all right. And then Alzheimer's disease, of course, a concern for the older folks. So how is the trisomy affecting the biology of human beings across the lifespan? Here's another multiomics analysis where we're looking at 22,000 multiomics features, transcriptome, proteome, metabolome, immune cell repertoire, and 400 participants. We'll let the computer organize these trajectories into clusters. And then you see things like B cells, for example, that we all lose with age. People with Down syndrome lose them faster, more pronounced. The amino acid serine is depleted over time, more so in Down syndrome. On the other hand, you have all the immune cells, say double positive T cells, going up with age, elevated in Down syndrome. An RNA that is a marker of wound healing and inflammation, ICAM1, going up with age, more elevated in Down syndrome. So now you can break cohorts into age groups and analyze the impacts of the trisomy in different life stages. For example, in kids, the proteome analysis reveals many proteins dysregulated, but they are mostly different than those seen in adults. For example, this protein CBR3, encoded on chromosome 21, is commonly elevated in both children and adults. But when you do an overlap analysis, you see many proteins that are either upregulated or downregulated in adults, different proteins upregulated or downregulated in kids. Let me give you 2 examples. Master regulator platelet biology, depleted in kids with Down syndrome, not so in adults with Down syndrome. This other protein provides the construct, the chitinase 3 like 1, elevated in kids with Down syndrome, not in adults with Down syndrome. You can do this at the pathway level. You see that some pathways, some signaling pathways are always activated in Down syndrome, like the interferon response. Others are always depleted, like complement. These 2 things go together. This is a marker of immune hyperactivity. These are the proteins in those signatures. But strikingly, some signaling pathways have biphasic behavior in Down syndrome, like mTOR signaling, elevated early in life, depleted later in life, right? So it is possible. It is possible with biospecimens to understand the effect of the trisomy at different life stages. The last challenge that I want to address is this issue of highly heterogeneous presentation of Down syndrome. Every participant in this slide is handling that extra chromosome in their unique personal ways. Each of them can teach us something new. How could biospecimens help us accelerate a personalized medicine platform for Down syndrome study and management? This is very real. Okay, let me give you an example. We just finished a clinical trial for 42 individuals that went on to take a JAK inhibitor for immune skin conditions. On average, half of them had thyroid disease, 40% have congenital heart defects, 40% have obstructive sleep apnea. But when you unpack those averages shown here, you see every one of them has a different profile, a different combination of co-occurring conditions. How could biospecimens help us unravel this complexity? Let me give you another example. This is a publication came out last year by Micah and Matt and colleagues, showing that not everyone with Down syndrome expresses genes on chromosome 21 the same way. There are different molecular subtypes of Down syndrome, helping us think that we may be getting closer to a personalized precision medicine. Let me show you some data from this paper. There are 126 chromosome 21 genes in the whole blood transcriptome. Yes, most of them are overexpressed, but every blue dot is a different participant, a wide range of overexpression for these genes. Look at this participant, 484, top overexpressor for this chromosome 21 gene, but not so for this chromosome 21 gene. Now, this other participant, 594, is not overexpressing TSPIR, but is one of the top overexpressors of GET1. So with this data, Mike and colleagues arrived to a clustering solution based on chromosome 21 expression leading to 3 molecular subtypes, 1, 2, and 3, who have very different characteristics. Individuals in the MS1 group are overexpressing genes involving cellular proliferation, like the oncogene NRAS, for example. Look at that. The 2 subgroups don't overexpress NRAS. Now, participants in the MS3 cluster overexpress many genes involving cytokine signaling, like the cytokine receptor, okay? Different flavors of Down syndrome, the molecular level. And you may be asking, what about the clinical profiles? At this sample size, there are some interesting trends. Participants with MS3 subtype have less of some conditions more than others. But when you apply false discovery rate penalties, you don't get a good Q value. Larger samples required to see these discoveries become of prognostic value. Which then leads me to another key area for which biospecimens will be needed. We need biomarkers in Down syndrome, right? Showing you this picture from a paper from Ida and Liz, where we have a very clear understanding of the biomarkers of Alzheimer's disease in Down syndrome that can then inform the therapeutic approach and the timing of the intervention, right? Even for universal hallmarks like Alzheimer's disease, there's great variability in presentation and severity. So the Alzheimer's Biomarkers Consortium Down syndrome is clearly addressing this. But how about other co-occurring conditions? Let me give you an example. People with Down syndrome are at a high risk of autoimmunity. More than 60% of them have autoimmune conditions. The most common is autoimmune thyroid disease. Then immune skin diseases like alopecia areata, atopic dermatitis. 10% have celiac disease, right? How about autoantibodies? They make a lot of autoantibodies. Just you measure these 2 autoantibodies, ANA, TPO, 80% of them are positive for at least one of them. Is this just the tip of the iceberg? What if you measure more autoantibodies? We did. Look at this. We measure more autoantibodies. Only 2 out of 120 participants didn't have autoantibodies. Look at all these autoantibodies elevated in Down syndrome in green, right? So now we can map autoantibodies to clinical presentation. If you see red, a red dot, it means a positive association. This is known. If you're making antibodies against thyroid peroxidase, TPO, you're more likely to develop hypothyroidism. But this is not known. Look at this. If you're making antibodies against this myosin protein, the electronic medical record says you are at a higher risk of a second neurological condition, right? Prompting the question, what are all the autoimmune phenotypes of Down syndrome? So the task order is very clear. We must collect biospecimens and associated metadata to keep driving these transformative discoveries, prioritize the resources, and eventually translate those discoveries into tangible solutions for people with Down syndrome. So the biobank, the biorepository, will be collaborating with collecting sites to facilitate collection of multiple types of biospecimens, both ante-mortem and post-mortem, with the sole exception of human fetal tissue. So we'll serve as a hub for expertise and collaboration in anything biospecimen related. We'll also partner with the collecting sites to develop common protocols for collection, processing, and analysis. We must make sure that every drop of blood is used to the maximum extent, right? To collect DNA, RNA, to use that plasma for all these wonderful omics studies, the white blood cells. For again, a multitude of studies generate cell lines, also analyze other cell types, right? We will also facilitate common collection kits to ensure that we are using the same materials, the same protocols, so that we'll drive collaboration through harmonization. And to wrap up here, how am I doing on time? I'm doing all right. I want to tell you a little bit about our vision for collaboration with collecting sites. 3 main collaboration modes, what we call full life cycle, hybrid, federated, virtual. Let me tell you more about this. Full life cycle means we'll be working with the collecting sites at every step of the protocol. We'll work together to design a biospecimen collection kit, send those kits to the site, get those samples back, store them, manage their storage and distribution. An example of this is the CDP, for example. This is how we are going to be working with the CDP. Hybrid sites, maybe sites that are already collecting biospecimens, but they may choose to partner with a biorepository for downstream steps, such as storage, processing, distribution. A couple examples could be maybe extraction of nucleic acids, generation of cell lines. There may be studies that have biospecimens that are going inactive, legacy studies that may donate the biospecimens for future generations through the DS biorepository. And then yet another mode of collaboration that we're calling federated virtual. There may be sites that are already collecting biospecimens and already doing the processing and analysis. So that we would like to work with them to collect the biospecimen catalogs to drive collaboration through the INCLUDE virtual biorepository. Examples will be the Human Trisomy Project, the ABCDS, the Down Syndrome Biobank Consortium, and more. Which leads me then into the virtual biorepository. What is this? This is an online platform to search, identify, and request biospecimens of a specific characteristics, right? And then depending on the agreements done with the various sites, you know, the governance maybe by the collection sites, maybe by the DS biorepository, there may be a shared governance. All of this will be, of course, agreed upon in material and data transfer and use agreements. To wrap up here, we already have a decent version of this virtual biorepository. You can, Adam earlier today demonstrated some of that. You can identify biospecimens with specific characteristics in the INCLUDE Data Hub. For the HTP, we have a second biorepository there in the Trisomy Explorer. You can ask biospecimens right now, set up collaborations, embark on new investigations. Now we'll have a beefed up version of this when we launch the website for the DS biorepository. Stay tuned, make sure that you're still subscribed to the DCC newsletter. So I want to end up here on a high note. I'm very excited. I think we should all be. There are more research participants with Down syndrome today than ever before. Look at that. 9,000 plus participants in the Data Hub alone. There are more Down syndrome researchers than ever before, thanks to the INCLUDE Project. 50 plus human research studies supported by the INCLUDE Project, 15 of them clinical trials. There are more clinical trials for Down syndrome today than ever before. And as Jessica explained, look at that. We're going to have about 3,000 more participants studying in great detail through the CDP. My last slide, my gratitude statement. I want to thank all people with Down syndrome and their families for keeping us busy and giving us something meaningful to do with our lives. I want to also thank colleagues across the various resources, the INCLUDE Project, the Data Coordinating Center, DS Connect, the DS4C, and then here at home at the Linda Sirnick Institute for Down Syndrome and the Human Trisomy Project. Everything we do at the Sirnick Institute, we do so in collaboration with the Global Down Syndrome Foundation. And I also want to thank other advocacy organizations. So I did rush through that a little bit because I wanted to make sure that there's time for questions for any of the presentations today. Thank you for participating today. Any questions, comments, or criticisms for any of the speakers today? Natalia is asking whether the recording of the session will be shared, and the answer is yes. We have a page in the Help Center with all the recording webinars.
Thank you. Thank you. That was great.
Thank you, Catherine. Who is that?
That's Mark's son, the littlest one.
Very good.
He's 8 months old.
Congratulations.
Okay.
So
Bye-bye.
Rafael is asking, what is the process for establishing a new group dedicated to biospecimen collection and research? Please reach out to us at the DS Bio repository. You can reach out to me directly, and we can start a collaboration. Again, we'll have an online presence coming up soon. You'll be hearing about it. In the meantime, please feel free to reach out. And Haley has posted my email address there. And Jean Ling Liu is asking about proposed sample size for the biospecimen collection. We don't have a limit on sample size. So if you have any study that would like to partner with a repository for 10 participants, that's good. If you have 300 participants, that's good too. We don't have a threshold for collaboration.
May I ask a question here?
Please.
Yeah, thank you for the great presentation that I've seen those. So my question was more like, do you have kind of what's the pool, like total sample size, you are proposing to have for the biospecimen?
Well, for the CDP, we're trying to get as much biospecimens as possible out of a cohort that may be in the 3,000, right? So that'd be very good. Now, there are other studies already collecting biospecimens. The Human Trisom Project has biospecimen on about 1,400 participants. ABCDS, the target number there is, I think, 500 participants. Those are all the adults. So various studies aim for different sample sizes. I think I may know where you're going in terms of, I know you're interested in genomics. So we have 5,000 genomes of people with Down syndrome in the hub right now.
Oh, wow.
Assuming, if I'm tracking correctly, I think I am, assuming some overlap with the CDP, we don't know at this point, how many of those participants will be in the CDP. I think it's safe to say that the CDP will add hundreds, if not a couple thousand new participants that are not in the hub yet. And you may have heard about a GUID strategy in previous webinars about how to find out whether a participant already got is a dual citizen of more than one study. So I think we may be in our lifetime, we may be looking at 10,000 genomes from people with Down syndrome. This is something I think in the horizon.
Oh, that's great to know. So you mentioned there are already like 5,000 whole genome participants.
Yeah.
Where? You mean either include the DCC or?
Yes, that's my count. Unless I'm wrestling or someone else corrects me, my count is nearly 5,000. Yeah.
Okay, awesome. Okay, thank you so much.
Very good. Very good. I want to thank all the colleagues at the Include Data Coordinating Center for the first few presentations. Jessica and Tracy for giving us an update on the CDP. We'll see you at the next webinar in the fall. Thank you very much, everybody.